Welcome to lesson 2 in immunity, and I'm going to be looking at phagocytosis. So if we have a look at the AQA spec here, and I'll just point out the bit that we're quite interested in for this video, and it's particularly about phagocytosis of pathogens and the subsequent destruction of ingested pathogens by lysosomes. So a phagocyte is a type of white blood cell, and there are many different types of white blood cells. Phagocytes in particular, like all other white blood cells, they have a nucleus and then all the other cellular substructures you would expect. And they are designed to engulf pathogens. So that's what we're going to have a look at and see how they engulf and then digest and destroy the pathogen. So firstly, the phagocyte has to locate the pathogen. If it's a bacteria, then it will locate it through chemoattractants, which are chemicals released by the bacteria, and it moves towards the pathogen along a concentration gradient. So it will move around, and I will show you a video in a second of that actually happening. And then once the phagocyte catches up with the pathogen, it then recognizes the foreign antigen on the pathogen and it binds. So the phagocyte has several receptors on their cell surface membrane that recognize and then attach to the surface of the pathogen, the antigens on the foreign antigens on the pathogen. So now the cytoplasm of the phagocyte moves round the pathogen and it starts to engulf it. And something called a lysosome, which you would have learnt about when we looked at cell ultrastructure. So lysosomes here, they then move towards the phagosome that is forming here by the engulfing of the bacterium. Now the phagosome is a form of a vesicle, and it's just known as a phagosome, but it is a vesicle. Within the lysosome there are lytic enzymes, and these start to break down the bacterium inside the phagosome. So the lysosomes, they will be destroying the ingested bacteria, and they'll be hydrolyzing their cell walls. And this is the same as what would happen for the digestion of food in the intestines. And some of those products are absorbed by the phagocyte, but some of the antigens, the pathogen's antigens, are then presented on the cell surface of the phagocyte. And this is important for the next stage of immunity, so the specific immune response. So what I'm going to do now is I'm going to show you a quick animation of what's going on, just to recap all of those points. Okay, so here we have the phagocyte and it's moving towards the pathogen, and you will see that it's attached to the cell membrane. Now, it attaches in a specific way, but you can see the receptors on the phagocyte and they bind with a complementary shaped antigen on the pathogen. What happens then is that the phagocyte will begin to engulf and form a phagosome, and then the lysosomes will go towards the phagosome and then release their lytic enzymes, and those enzymes, those digestive enzymes, will then start to break down the pathogen and destroy the cell walls, and therefore making it inactive, and then they will absorb some of the products, and then the antigens will be displayed on the cell surface. And finally, here's a quick overall summary that you might like to pause and label the parts in the revision booklet. Okay, so that's the end of this lesson for looking at phagocytosis. So next lesson we'll be looking at the specific immune response.